Our first seminar speaker today is Dr. Helena Morrison, and to introduce her is one of our former UBRP students, Rebecca Gardner. So, Rebecca, would you come up? Hello, everyone. So I'm so honored today to be able to introduce to you my mentor and friend, Dr. Helena Morrison. Dr. Morrison earned her bachelor's degree in animal science at the University of California, Davis, a bachelor's in nursing at Angelo State University, and her PhD in nursing at the University of Arizona. She's a registered nurse and is the principal investigator of a lab within the College of Nursing, whose research primarily focuses on, but is not limited to, ischemic stroke, neuroinflammation, sex differences, microgliomorphology, and neurological diseases. She was also nominated and awarded the 2019 UBRP Outstanding Faculty Mentor Award at this past UBRP conference. Not only is Dr. Morrison enthusiastic about her research, but she's also an incredibly fascinating person to learn from and a great role model to have. If any of you have questions at all after her talk, I strongly encourage you to take the time to stop and ask, because learning from Dr. Morrison is truly a privilege. And with that, I'm happy to hand it over to Dr. Morrison.
Well, that was such a nice introduction. It's actually a privilege to work with all UBRP students. I have a long time been working with UBRP students since I got here in 2014. It's actually, I tell this to Becca all the time, it's the thing I actually like about my job. Most of the time, when things are really tough and I'm not really enthusiastic about what I'm doing, I love coming to work and working with undergraduate students and UBRPers. So we call you all sometimes. So the title of my talk, if you all want to know about all the science that I do and the nitty-gritties, you can read my papers, you can watch my Joe videos. There's lots of detail in there. I'm going to show you some pretty pictures and talk about some of my science. But one of the things I also wanted to talk to you about, since this is such a mixed group, is how I got to where I am today. Because I think it's fairly, at least when I was a student, an undergraduate student, I saw my professors and I was like, wow, how did they get there? And I'm going to tell you how I got here, because it's a path that is not very common, but it put me in a very unique position to contribute to science and to a profession that I loved, and that didn't know that I loved right away. So the title of my talk is Be a Nurse and a Scientist. Some people think that it's a mutually exclusive categories, but they're not. And my path that evolved from suggestions and the desire to know more. So yeah, truly, I am here today from a few people that weren't really important to my life, actually, that made some suggestions that I thought about and I took. So I'm going to tell you about that. I come from a family of nurses. My grandma was a psych nurse in like the 60s and 50s. Like that's not a great time to be a psych nurse back then, because I'm pretty sure some of those movies are correct with, you know, cages and strapping people down. But that's what she was. She was a psych nurse in the 60s. I have her diploma in my office. I think it's really rad to look at often, to know where my roots are. My mom was a nurse, a type of a nurse. There's lots of different nurses. That's a whole other talk. My aunt just retired as a nurse, working for an OBGYN. So I have a strong predilection to go into nursing. And so I thought for the first 10 years of my life, I'm going to be a nurse. But then I learned about animals, and I'm like, wow, I like working with animals more than people. So I had a deep dive out of that trajectory. I went to UC Davis, as Becca mentioned. I graduated from animal science, and I powered through that, finished in under 4 years. Thought I knew what I wanted to do. I started the degree. I was one of the few people that I knew of that entered as a declared major, and like, I love animal science, I'm going to do this. I didn't want to be a vet. I kind of learned that really early. And I had made up this profession in my mind of what I could do with an animal science degree. Well, that was, like I mentioned, totally made up. As it turns out, what I thought I could do in my head really didn't exist. Didn't really explore my professional opportunities while I was in school, because I was so busy just trying to get finished and get graduated. I thought I was going to be a farrier for a short period of time. I don't know if does anybody know what a farrier is? Yeah, you shoot horses. So I was going to do that. really glad I chose not to do that, but I do have some amazing horse trimming skills and I actually know how to shoot horses to some extent. So I graduated with my animal science degree. I went out into the big wide world and went, wow, you can't really do anything except work for vets, but you don't need an animal science degree. But I did get married right out of school, which was a motivating factor to get finished with school. And I joined the military by de facto. I married a military guy. So one of the benefits that was available to me, and this is take advantage of every available benefit and resource, was career counseling. And this random person who I met once in my life said, why don't you become a nurse? Because it's a really marketable degree. You can move every 3 years and get a job. And at first I was extremely irritated because here I have a very highly marketable, in my mind, animal science degree, and this guy's telling me to go do something else. And I thought about it, really, after my knee-jerk response of no, I thought about it, I'm like, well, but wait, I wanted to be a nurse, like, back then. Why don't I just go back and do that? It's a lot harder than it thinks. Nursing is a certified program. Their curriculum is very regimented because of this certification, so there's lots of hoops, and location is always a difficulty. But by and large, I ended up getting my LVN, which is a certification, kind of. So I kind of demoted myself back to a 9-month degree. Then my associate's degree in nursing, which has enabled me to sit my RN state licensure and practice nursing as an RN. Went straight in to get my BSN through a cobbled-through program that was kind of like ABN and BSN. It's supposed to be fast track, it wasn't very fast. And during that time, I was able to practice nursing, and I loved it. So I learned that I loved it right away, and that was awesome. I worked as a med-surg nurse, postpartum, oddly enough, ER, and then my passion was intensive care nursing. So it was there that I had the drive to to know more. That's where the know more comes from. I didn't want to just be a consumer of knowledge, and that's what the practice of nursing is. You take the knowledge that has been developed by others and you implement it at the bedside to take care of patients and hopefully prevent them from dying, or at least help them get better. And so that's what I did. But I realized very quickly that that wasn't enough for me. I wanted to be a person that developed that knowledge base. And I wanted to move practice forward, and I wanted to move therapies forward for the stroke patients that I was taking care of that there really were no treatments for. All I could do was put them on a ventilator and pray that their brain healed itself. So, I went on and got my PhD here at the U of A, actually, because I talked about suggestions. There was somebody way back here in my UC Davis years, ran a postdoc. I was a TA for one of my small classes, who was a computer scientist and doing a postdoc in animal science. And I thought that was really weird. Turns out he was modeling ruminant digestion so that he could build a computer model back in the days. This is like Apple IIe's, floppy disks. Okay. So that he could model ruminant digestion, which is highly dynamic. We needed calculus to understand the rates of digestion of a 4-chambered stomach, so that what you put into a cow, he could better predict what came out in milk quality. Feed them soybeans, get a higher protein content, these types of things. So he told me as I was sitting in class one day, you know what, Helena, the key to success is not following the path that everybody takes, it's finding the niche. It's finding the thing that nobody else does, because we can all compete to be the best of the best of the best, and that's great. But you can also compete to fill a niche that nobody else has the skills to have. And so I think back on that still today. What is that niche and how can I fill it? And I do that for nursing. I'm a nurse physiologist. I combine my passion for animal science and the nitty-gritty details of learning how cells and systems work with my nursing clinical background. There's maybe about 6 to 10 of us nationwide with that particular set of skills. So it makes me highly sought after with lots of different types of positions in the nursing profession. So I studied neutrophils, which is a type of phagocyte that's actually in the systemic circulation, and how that interacted with the brain to increase brain injury post-stroke. And you'll see why that's important in just a minute, where after I graduated in 2005, I did a postdoctoral fellowship in a physiology lab, and they all forgot that I was a nurse until even though I keep telling them, and I signed my name with RN at the end of it. Until I got a faculty position, they're like, wait, you're a nurse? Oh, yes, I am a nurse. And I do bench science. But it was there that I learned to look at brain cells, not systemic cells, blood cells, and to learn how brain cells are reacting post-stroke and how these cells contribute to either recovery or exacerbation of injury after stroke. And I mentioned the neutrophil here because the twin sister of the neutrophil in the brain is the microglia. They're also phagocytes, but they're resident. They're already there. They don't need to infiltrate because they're these branchy, highly ramified cells. We're talking about those in just a minute. So that was my path to getting to where I am. In 2014, I joined the faculty at the College of Nursing here, and here I am today, 5 years later. My take-home points that I constantly remember as I move through my career, and I try to remind students as they're navigating their ambitions and trying to get out of school at the same time. Find your niche, listen to suggestions. They may seem random, but they may come back to you at some unknown time and have great meaning in your life, even though you've never seen that person before. So just consider all the suggestions that are open for you. Do your homework. Find out if that job actually exists. That's easy to do. Shadow people, interview, whatever. Look it up on the internet. We didn't have the internet when I went to school, so you guys have that for you. And then just persist. This was a not very linear, direct pathway, and I was older than a lot of my colleagues, but persistence counts, and I'm doing what I love today and living a dream. So go do it. Okay, so the bulk of my research, and I'm going to tell you a little bit more broadly about some of my research, is I study brain cells to develop precise and personalized stroke therapies. At least that's my ambition. And these are a bunch of those different brain cells. These are microglia. The blue ones are astrocytes, and believe it or not, the red is not a vascular stain. This is immunohistochemistry if some of you don't do some of this type of work. The red stain here is actually a type of receptor that's on astrocytes that are on the interface of the vasculature. They wrap their end feet wrap around the vasculature, and so because I stained for that receptor, it looks like a vascular stain, but it's really part of the blue cells that you see here. So these are all the cells that I've learned to study over the years in the brain, and I study ischemic stroke because there really are no therapies for stroke. That said, when I first started studying stroke in 2010 as a graduate student, it was the third leading cause of death. It is now no longer the third leading cause of death. It is like the fifth or sixth, depending on the year. You could think that we've gotten better at treating stroke, but I just told you we have no stroke treatments and no novel therapies, right? So it's actually not, as a system, as a hospital system, we have gotten much more efficient at treating stroke. Our patients are much more aware of stroke symptoms. They're getting to the hospital in the 4-hour time period that we have to deliver the one therapy we have, which is called, we call it a clot buster, it's TPA. All it does is break up the stroke, the clot, and reperfuse the brain. That's it. We have no neuroprotectants. We can't decrease inflammation at this point. All of these problems that accompany stroke, there are no therapies. Studying stroke is very difficult. It's not widely popular right now. Much more popular diseases are neurodegenerative diseases such as Parkinson's and Alzheimer's, so it's always a struggle to get stroke funding. And a lot of that is because we have failed time and time again over the years to translate potential therapies into stroke. So, one of the reasons why, and I'll talk about this in just a few minutes, one of the reasons why I love to collaborate is because sometimes the thing that you're happened to be studying is really difficult. And it's like beating your head against the wall. But if you can collaborate and work with other people on their studies and be an expert in one small segment of their area of study, their stuff may be working great. And that is a great motivator even when your own independent line of research is sometimes a struggle. So it's great to network in science, to work with other people because it gets you through the tough very true that 80% of strokes can be prevented. The comorbidities for stroke are all non-modifiable. Many of the comorbidities for stroke are modifiable, lifestyle-oriented, and hereditary issues. Diabetes, hypertension, AFib, these are all things that lead to stroke. But one of the things that is non-modifiable that interestingly affects stroke is sex and sex differences. We do know this, and this is about all we know. We know that age is the other non-modifiable risk for stroke. As you get older, your risk for stroke gets greater, and the severity of that stroke gets greater. But for males, simplistically speaking, this relationship is fairly linear. For females, it's a little bit, the slope is lower, so they're protected during the early premenopausal part of their life. But at postmenopause, all of a sudden something changes. We don't know what exactly. And this relationship changes and becomes much more severe of a slope. So by the time the average age of stroke, women have a greater stroke size, and it impacts them functionally much more robustly, or maybe much is a little too much to say, but more robustly than their age-matched male counterpart. So although we know this to be true epidemiologically, we are still studying the mechanisms of why this happens. As you can imagine, a lot of people think that it's sex hormones. There's also sex chromosomes in there. Developing models to study that can be quite challenging and quite extensive. It's easy just to study males because you have like one set of group and you only need like an ANOVA or a t-text. But if you start adding sex into that equation, you have doubled all of your work. And if you think that hormones have something to do with it, you now have females that are estrus, diestrus, which are different levels of sex hormones, as well as postmenopausal, which happens to be the most important part for stroke because that's when strokes occur in females. So that, if you want to study all those things, your 2-group study now just became a 4 or 5-group study. So I can understand why a lot of people don't study sex differences, even though the NIH wants us to do that. Biological variable that I do not study because I don't have room in all of my groups is age. I separate because another wonderful investigator at the U of A, through my collaborations I learned, developed a model to develop postmenopause in age-matched females. So I don't have to wait for these females to get very, very old and, by the way, fragile, to study them in a postmenopausal state. I can induce postmenopause in my mice and age match them to 16 weeks old. So take age out of the equation so it's no longer coupled with postmenopause. And therefore I'm only studying the effects of the postmenopausal status on the system. And so yeah, I'm really lucky. That's one of the other reasons why I came back here to U of A. So this is like a summary. It's really hard to see, but what I want you to see, I don't want you to really read anything. These are kind of the relationships of the vasculature. So this is what gets blocked in the brain. There's like a big bowling ball here, okay? And all of these cells are associated with this blocked vessel that's right here in the middle. When I started at U of A in my graduate studies, I looked at immune cells in the circulation, neutrophils, macrophages, things like that. It does come full circle, I'll tell you. Becca's actually helping me work on a project that helps distinguish microglia, because again, they're the twin sisters, so it's really hard to distinguish between them, and the infiltrating blood cells, macrophages, that happens when blood-brain barrier breaks down and some of these external cells get in. We have no tools right now to distinguish these two populations except for morphology, which happens to be my specialty and my contribution to a lot of the science that happens in this area. I also study astrocytes, and these are what I was talking about, the end feet that wrap themselves around the vasculature. So these cells in combination have a good, bad, and ugly side of them. One would think, well, okay, if these are bad or something, let's just get rid of them and then it'll all be good. Well, there's two sides to every coin. You'll find this common theme throughout your study of the body, if that's what you happen to be studying. Whoever made us, whatever made us, did it efficiently so that we can have the same thing doing two different things. Unfortunate for us, if we're trying to intervene on that, you have to do it at the right time, at the right place, at the right location, so you're not throwing the baby out with bathwater. You do not want to get rid of the good, although it seems like there's a lot less good here than there is bad. As scientists, we always like to study bad things. I know that because I do like studying good things, and it's hard to get people to fund that. So we have to find out the very precise mechanisms to get to lessen, to dampen some of these curves, or promote some of these curves so that it can balance this out. So that's our goal with studying these types of mechanisms. And that's why we have to be very precise in how we go about doing it and in the research questions that we have in our experimental design. So, I wonder if they're going to play. These are movies that I took. So microglia are the brain immune cell, and they are highly dynamic cells. It's going to work. I took videos of these in acute brain slices, and their processes actually reach out and touch their surrounding. They're the surveyors of the brain. If I'm a microglia and like you're a neuron, I'm going, hey, how you doing? How you doing? And you're telling me back through communication, cell messaging, and things like that. I'm fine, I'm fine. And so I just go about my business, and these processes survey the brain. It's estimated they're highly active once every couple hours. Okay? So if that neuron, you as a neuron, stop talking to me because all of a sudden you're ischemic or something, a lot of times I, as a microglia, will hone in and go, what's going on, what's going on? So you can have an increase in process activity. You definitely see a change in morphology, and that's something that I specialize in studying, like I mentioned. So if you have a very ischemic, this is like all in dead tissue, you can see these cells have transformed themselves into these much more amoeboid. Their processes have retracted. It's called deramification, and they now turn into full-blown phagocytes, not just pruning cells, but they're nice, finely tuned processes. But now they're just like, give me all your dead. It's like the Monty Python movie, you know, bring out your. All right, so yeah, that's the response. So if we can finely tune this, because they're turned on very specifically, but once they're turned on, they just go to town. So they're eating dead stuff, they're eating live stuff, they're eating stuff that if you just like promoted their health and wellness, they would survive. So if we can figure that balance out, then we could save brain cells, right? And if you save brain cells, you save brain function. So that's the goal with studying phagocytosis. It's not easy, but we aspire to it. So like I mentioned, there's all these different types of ramification, cell morphologies. This is what a typical stroke looks like in my lab, which I deliver to mice in a very controlled way, so that it's, at least that's controlled. You have these ramified cells that we have through imaging, processing, and things like that. We convert them into binary skeletons. And then there's awesome free software out there that is used to quantify some morphologic aspects. Once we have built, this is basically model building here. And so I'm able to track then by doing this in this brain region, in this brain region, in this brain region, in this brain region, I can see how the evolution of the morphology changes in proximity to the severity of stroke. That had never been done before. Most of the time, right before I started doing this, in my postdoc days, people would literally, in literature, point arrows to cells and go, this one's deramified. So categorical data analysis is not nearly as precise as other types of analyses. So I need to develop a method to do a more continuous variable, and this is what I came up with. So again, this demonstrates in the process of phagocytosis, they change their morphology, and they start eating stuff and getting bigger. So in my lab, what we investigate, my independent line of research is with ischemic stroke. But because I have expertise in studying microglia and I know their wily ways, I collaborate a lot with other people. Most productive collaborations have been in traumatic brain injury. It's very similar to stroke, slightly different reason for a brain injury. And also Parkinson's disease. That would take me a while to go into, so I'm just going to leave it at that. These are the methods that I use. The postmenopause model, ischemic stroke model, that's me at the surgery table delivering a stroke. and immunohistochemistry, confocal imaging to take these pretty pictures. We do a lot of histological analysis through imaging. I also do a lot of protein work when I have to, using multiplex and Western blots, and I actually am skilled in flow cytometry as well. So we actually, when we need to, we have a large capacity to answer a lot of different questions. Right now I have a small lab. Susan Whitman is my part-time technician. Becca here is in the lab, and if you don't get a hold of me, you can always talk to Becca, I'm sure. And then a nursing student, Victoria, who is helping me with some data analysis. So, if you have any questions about nursing, nurses who do science, my laboratory work, feel free to contact me at any point, email me, whatever's most easy for you. That's about what I have. The neurons do not turn into phagocytes. The microglia are phagocytes, and they start eating neurons that are dead, that are around them. And it's their job to basically monitor them. I call them, they're the brain's first responders. So actually, I'm technically trained first responder as well. So they are the first responder to brain injury, or even increased brain activity. I also have been involved in some epilepsy studies. So there, you just have increased neuronal activity. They're not dead, but they're aberrant activity. And the microglia look drastically different because they don't know what to do with themselves. They're getting all kinds of signals from the neuron here, and their activity increases quite a bit. I don't. The only time I get to work with patients, I do have some nursing graduate students, and very few of them don't like to do bench science. And so most of them are doing their graduate work in patient populations, and that does bring me back to the bedside. I have a student right now that we are doing chart reviews to look at some association between biomarkers and comorbid conditions. But I don't practice at the bedside anymore. Science is a full-time job. Bedside nursing is also a full-time job, so they don't like it when I call out I learned that early on from a 12-hour shift because I have to repeat my experiment because it didn't work the day before. I tried that in grad school, it didn't work very well.
All right, thank you again. To introduce Dr. Arnold, I'd like to invite up Victoria Howard. Oh, and I was also looking for Jasmine. Oh, there you go. Cool. I'm honored to introduce to y'all today Dr. Betsy Arnold. Dr. Arnold is currently a professor of plant science and ecology and evolutionary biology here at the University of Arizona. She is incredibly active in research in fungal ecology and evolution and is strongly dedicated to mentorship, teaching, outreach, service, and fostering diversity in STEM. She obtained a Bachelor of Science at Duke University and then worked at the Smithsonian Tropical Research Institute in Panama. She then earned her PhD as a National Science Foundation Graduate Fellow here at the U of A, and then completed an NSF postdoctoral fellowship at Duke University in microbial biology. In 2005, she returned to the U of A and has since mentored a diverse group of postdocs, undergrads, graduate students, high school students, Indian, tribal college students, and many more. Her research is internationally recognized, and she's also an award-winning teacher in microbiology and related fields. I'm tremendously grateful to have her support and mentorship throughout my time at UBRP. So without further ado, here's Dr. Betsy Arnold.
So Victoria, thank you so much. That was awesome. I'm just thinking that when I was an undergraduate, I would have had a really hard time giving an introduction like that. So extra round of applause. That was really good. Yeah, so I'm really glad to be here, and I want to thank all of you for being here today too. I'm really excited to tell you a little bit about my research. And obviously, it's a bit of a transition from the last talk. So everybody get ready to change gears. And please take a moment and take a look at the slide that you see up there. And think a little bit about the organisms that you can see there. And think a little bit about the organisms that you can't see. And hopefully by the end of the talk, you'll understand my appreciation for this little piece of nature and for the literally thousands of species that are present here. Okay? And this is an area that's about this large. Okay? All right. So the title of my talk is Fungal Diversity and Applications, Perspectives from a Hidden World. I just thought I'd start by asking, how many people like these things? Anybody? Yeah, and how many people want to stay as far away from them as possible, not on your pizza? Yeah. Oh, Victoria, really? Yeah. Yeah, so it's one of those watershed foods, right? So what we're looking at here are the fruiting bodies of a fungus, right? And if you're normal people, which I think everyone is, you didn't wake up this morning thinking about fungi. I did, because I knew it was going to be this awesome monsoon day. It's a great day for fungus. But when I'm thinking a little bit about why fungi are cool, I don't stop with this bowl of mushrooms. This is like the iceberg lettuce of the fungal world. Okay? And I like iceberg lettuce, don't get me wrong. But actually, what's really great about these is that fungi as a whole are incredibly important in human endeavors. They are the reason we have beer and wine. They are the reasons that bread rises. Fungi, of course, are on our mushroom pizzas. So we can eat some fungi or fungal products, the CO2 that's evolved from yeast in the process of baking the brew. Fungi are also cool because they're eukaryotes, right? So we're not talking about bacteria or archaea. We're talking about members of our giant branch of the tree of life. And in fact, fungi are more closely related to us than they are to plants. Okay? So these are like our fungal cousins that we should appreciate as maybe our distant but important relatives. Fungi are also heterotrophs, and so I'm sure that's not a new word for many of you. An autotroph is like a plant, it makes its own food. A heterotroph has to eat something else. So the great thing about fungi is that they grow into their food and exude enzymes and then move those molecules across their cell walls, which are made of chitin, into the cells, and they can survive. So whereas we as animals have a chamber where we put our food and digest it, they basically are in a living bathtub of delicious food, and they're just growing into it and digesting what's around them. And what that means is they've got a ton of really awesome enzymes, and those enzymes can be applied in all sorts of areas: bioremediation of oil spills, recovery of degraded soils, industrial applications for recycling, and so on. And then finally, I appreciate fungi because this is a branch on the tree of life that's over 1 billion years old. So it is ancient and really diverse. Okay? And so what do I mean when I say fungi are diverse? Right? Well, actually, at the moment, we've only described about 100,000 species, give or take. And so what does it mean to describe a species? That means you put a Latin name on it, you describe what it looks like, you use some fancy language that talks about what it looks like on different growth media, under different temperatures, and you put it into a collection. So people can always go back and see that example. That's how we describe a species. Now, obviously that takes a lot of effort, and there are a lot of fungi out there that we haven't described yet. And so that's one reason that I, as a mycologist, someone who studies fungi, loves to work on fungi. Because we've, described 100,000 species, that's kind of a boatload. But we think that that's only the tip of the tiny iceberg. And in fact, there's a tremendous amount of biodiversity out there. So when we start to step out of our little beige mushrooms in the grocery store, we start to appreciate sort of this beautiful biodiversity. And once again, this is but a tiny slice. So we have earth stars, we have things like boletes or porcinis, morels, absolutely delicious. They tend to fruit after fire in forests, really amazing. In fact, I know families who have like secret morel hunting grounds that are passed down as secrets in the family because the mushrooms are so good. And then the sort of flagship one over here that was famous because it was one of the few fungi ever to be explicitly shown on National Geographic cover. Of course, it had a tree frog in it. I don't know, it was probably the mushroom, right? A really beautiful tropical mushroom. many of the fungi that are out there in the world look like these. These are the fruiting structures where spores are made. But the fungi that represent the greatest biodiversity and the ones that excite me the most are the humble ones you see up there. This is the stuff that you find growing on your bread or on your strawberries in the back of the fridge. These are the molds. These are fungi that don't have macroscopic fruiting bodies. They have microscopic fruiting bodies. And so we call them microfungi. Okay? So basically, what is a microfungus? It's a eukaryote that is a heterotroph that has microscopic fruiting bodies, and it's a member of the fungal lineage. Okay, so we summarize that by saying mold, and occasionally I talk to my mom about my work, and she's like, wow, you went to school for so long to study mold. I did, but you'll see why. I think it's really cool. All right, so when we say that we've described about 100,000 species, we've gone through that process for lots of the big fungi and lots of the little fungi. But actually, fungi are thought to be what we refer to as hyper-diverse. That means really, really diverse, tons of species out there. And in fact, the working estimates right now are that there's somewhere between 1.5 million and 9 million species of fungi on the planet. So for someone like me, that's totally like job security, right? There is a huge gap between what we've described so far and what we think is out there. So the question is, where are all those missing fungi, right? So the fungi between what we've done and studied and what we think are actually out there. Well, it turns out that the vast majority of fungi that are yet to be discovered are thought to be cryptic microfungal symbionts. So cryptic, hard to see in nature. Microfungal, you already know, producing microscopic fruiting bodies. And symbionts living in close association with another organism. And in fact, most of those are living in close association with plants. Okay? They're hard to see, okay? And they represent a tremendous richness of species. Okay. So what I'm doing is I'm setting the stage to tell you about the specific group of plant-associated fungi that my lab and I study. Okay? And so when we think about plant-associated fungi, everybody pause, picture your favorite plant, whatever that might be. I'll bet you. Some people it's Christmas trees, other people it's that iceberg lettuce. Some people are not thinking about plants. Plants, let's go. Okay. All right, so think about plants. So the thing about plants is that very much like we have a microbiome, okay? All of us are a bacterial habitat and fungal habitat. So too do plants have microbiomes. Very much like we get sick due to the effects of pathogens, so too plants can get sick. Okay? So plants have microbiomes, and the major component of plant microbiomes is fungi. For us, our microbiomes are predominantly bacteria. Okay? All right, so when we think about what kinds of fungi live with plants, the first ones are what we call epiphyllous fungi, or epiphytic. Epi means on, so this is on plant surfaces. You can see them, for example, walking through a tropical forest. It's white splotches and kind of rotty, nasty stuff on plants. Okay, I've already used this term pathogens, organisms that cause disease. Often we see evidence of those with brown spots on leaves, that sort of thing. And then for those who spend some time maybe in ecology or thinking about fungi or microbiology, you've probably heard about mycorrhizal fungi. These are fungi that live in association with roots. They're present in more than 80% of plant species out there, and they're really important for helping plants take in nutrients in the environment. Okay. So those are the three main groups that have been recognized for a long time in plant-associated microfungi. But actually, there's another group, and that's the group that my lab and I study. And it's a group called endophytes. Okay? Endo means inside, phyte means plant. So these are fungi that are occurring within plant tissue. And it turns out that we can actually define them very simply. Okay? Endophytes are fungi that live within apparently healthy plant tissues without causing any overt or clear harm to their hosts. Okay? So if everyone looks out the window there for a minute, take a look. You see some green leaves out there? And imagine each of those leaves as having inside it a fungal symbiont, and that symbiont not causing harm. Okay? So for us, that's a very exciting insight, because it means that wherever we look and see plants, there's a hidden world of fungi inside. And those fungi are endophytes. And in fact, when I go to places like this in the tropical forest, even though I'm in a plant sciences program here, what I love to do in my mind is strip away all that distracting green plant material and think of the beautiful endophytes inside. Okay? And that biodiversity really motivates me, because endophytes are a very poorly known group, yet they occur in every single plant that's ever been looked at anywhere in the world. So it's this vast reservoir of biodiversity. Okay, So I'm going to give you a one-slide history lesson on how we got into endophytes in the first place as scientists. So this is the poster child in endophyte biology. This is a cool season grass. Okay? So we see a beautiful grass plant here in our herbarium preparation. So it turns out that cool season grasses have endophytes. And this is where endophytes were first found. And there's some special traits that people thought maybe explained all endophytes, but that actually isn't the case. So in cool season grasses, endophytes are not very diverse. They represent only a single family. For anyone who likes fungi in the room, they're in the Ascomycota and the Capsicitaceae. Yeah, no one's like, yes, I love that family. And so the endophytes here, it's basically one endophyte species per plant. They are vertically transmitted, and that's a term that we use in all of microbiology. Basically, it means that the microbe passes from the maternal host to the offspring. Okay. We also know that they grow systemically. That means throughout the body of the grass. And then finally, they produce a bunch of important secondary metabolites. So secondary metabolites are compounds that the organism doesn't need to go through to have just for basic life, but give the organism a competitive advantage in certain situations. Okay? And so that's how these endophytes were found in the first place, actually. Back in the early 1900s, people realized that livestock consuming certain grasses would get sick, they'd get confused, they would lose energy. People thought that it was the fault of the grass. And in fact, there are grasses all around the world that have common names like drunk grass, or sleepy grass, or death grass. It turns out that it's not the grass at all. It's actually the endophyte in the grass that produces an alkaloid to which mammals are sensitive. Okay? So endophytes were discovered when people realized that livestock were getting sick. They started looking to see what's going on with the grass, and there were fungi inside. Fungi were isolated out, alkaloids were identified, and we understood clearly that it was the fault of the fungal-plant interaction. And this brushes up against human history in another way. It's actually thought that the Salem witches who were women several hundred years ago in New England who were criminalized for their behavior. It's thought that their behavior may have been caused by alkaloids from endophytes in the grain that they were grinding for bread. So they were basically on an LSD trip, is the idea. And so endophytes really can be important, but they're not just in grasses, and of course not all plants are grasses either. And so this is just a little clip of my field site in Hawaii. You can see we've got trees and ferns and all sorts of non-grass things. And when I started my work, going all the way back to the mid-90s, makes me very old, there was basically very little known about endophytes in these other plants. And what got exciting to me was that when I was 21, and I had started to think about how plants get along with other organisms, I came across this paper, and it changed my life. Okay? Now we think like, I don't know, like superhero origin stories are a little more dramatic, but no superhero here, just appreciation for the literature. So let me tell you why this is so cool. So what we're looking at here are pieces cut from a single leaf of a tropical tree. Okay? So that leaf has been cut into 4 big panels, and each of those 4 big panels has been cut into small rectangles. Okay? Each of those small rectangles is about 1 millimeter by about 2 millimeters. So if you make a little diamond, pinch your fingers together, it's about that big. Each different pattern is a different fungus. So basically, these leaves are these mosaics of biodiversity. As you move 1 millimeter to the next, there's another fungal species. And it's not just a different species, it's a species on a branch of the tree of life that may have been separated for millions of years. Yet it's in this healthy tissue. So this raises the question, how many species are capable of this friendly lifestyle? How many times has that evolved? And how can we capture the diversity of these friendly fungi, some of which are actually quite beneficial for plants? Okay? So those are the big questions, basically, that my lab studies. We try to scale up from this paper by Jean Lodge in 1996, basically showing in one leaf, to a global perspective where we're thinking about distributions of the fungi, how have they evolved to not be causing disease, what do they do, and how can we use them? Okay. All right, so with that background then, I just want to tell you a little bit about the work that we do in that context. And so one of the great things about fungi is they are everywhere. Anywhere we go around the planet, we can collect plants, we can evaluate their endophytes, and discover novel biodiversity. And so I just want to zoom in on where we have done most of our work, but as I'll show you at the end of the talk, we now have a much more global perspective. So over the last 20 years or so, we've sampled in the places marked with stars here. And I'm going to walk you through the process of collecting these endophytes so you get an idea of what we do. Okay? So the first thing I'm going to show you is an evolutionary tree. And this is an evolutionary tree showing the relationships of green things. Okay? So plants. If you don't know the names, don't worry about it. At the bottom you see algae. Everything else is either a non-vascular plant like a moss or a vascular plant like the trees we see outside. Okay? All right, so we have not only sampled basically from the tropics to the tundra, but we also have sampled across this entire tree of life. So trying to capture as many different hosts as we can. So the stars are corresponding to tropical, temperate, and boreal, and you can see the number of species in each lineage. Okay. So what do? Basically, we're like this hotshot team of microbiologists. We will fly into a site with our portable sterile hoods. We get out of our plane or helicopter or boat or car. We head to wherever we're going to collect. We then will set up transects, and we choose groups of plants that represent the local diversity of plants. We repeat that 3 times in a site to get a nice sample. What we do is that we take leaves that look healthy, and we subsample those in those little 1 by 2 millimeter pieces like you saw in the mosaic. Okay? We surface sterilize them. That's basically just washing and bleach and ethanol to get stuff off the outside so that when we put these in culture, we're only looking at the endophytes. Okay? When we do that, it basically looks like the photo you see here. We have a little piece of leaf growing out an endophyte on a growth medium. Now, one thing that we know is that lots of endophytes can't be isolated in culture. For those of you with a background in microbiology, you know that many microbes can't be cultured, right? It's true for endophytes too. But our lab specializes first in coming up with creative ways to culture, so we get the greatest possible biodiversity in our collection, and then we use next generation sequencing, typically on the Illumina platform, to capture what we can't isolate in the culture. Okay. I'm going to talk about mostly cultures today because that biodiversity really motivates me. Okay. So typically we will culture our plant material within 48 hours, and that's why in remote locations it helps to have our little portable sterile lab. Just get right to it. And the reason that we do that is that if we keep the plant material around too long, basically the fungi that are good at breaking down plant material grow really well, but the symbionts that are really dependent on that living material don't tend to survive very long. So we really have to act fast if we want to capture that biodiversity. Okay. All right. We always make sure that we're doing controls, and we're not getting stuff off the surface, but we're looking inside. And then we'll often incubate these little 1 by 2 millimeter segments, thousands of them, for at least 1 year. And let me show you why. So I'm going to show you a graph. And what we see here is the percent of endophytes that we get from a collection as a function of how many months we've had it in culture. Now you can see if we just want numbers, we could cut it off after 1 month, right? Because we get tons of stuff growing. But what's awesome is that if we leave those pieces around over time, even though we don't get a lot of cultures coming out after 11 months, those tend to be biochemically really interesting, producing very unusual secondary metabolites, or they represent interesting evolutionary lineages. So it's worth it, and luckily we're often overwhelmed by how much we're culturing, so the stuff sits there for a year anyway. So it kind of works out pretty well. Okay. All right, so I'll just close this section by highlighting this box at the bottom. So I told you that we have green algae, right? So where do we find green algae in terrestrial environments? We find them in the context of lichens. So many of you who spend time outside have seen these crusty, colorful growths that are on rocks and trees, especially in wet areas. And lichens, okay, are well known as a symbiosis between a photosynthetic partner, an alga or a cyanobacteria, and a fungal partner called the mycobiont. For a long time, scientists thought there were 2 partners, photobiont and mycobiont, end of story. Turns out that if you take these lichens, like we see here, you surface sterilize them just like a leaf, cut them up and let stuff grow out, you get lots of additional fungi. And we refer to those not as endophytes, but endolichenic fungi, because they're inside lichens. Okay? And so often we find them in association with those photosynthetic cells. So when you take all of the sampling that we've done, what it does is it creates a globally unique biodiversity collection that's housed in my lab here at the university. And this is the largest collection of these organisms on the planet, and we're very proud of it. And I think they're beautiful. To me, that's more beautiful than looking like at a stained glass window in a church. In part because the vast majority of the species that we see, here just a tiny sample growing on petri dishes. the vast majority that we see are new to science. They're species that no one has ever seen before, because who would look in healthy plant material? Okay. So what we do with our collection is that we currently have more than 80,000 of these species living in our lab. We sequence a part of their genome that is basically used to identify fungi. It's part of the nuclear ribosomal repeat called the internal transcript spacer. We also will sequence various protein-coding genes to look at function. And in some cases, we'll sequence the entire genome. The genomes are about 40 to 50 megabases, so pretty standard for fungi that have a free living stage as well as the symbiotic stage. Okay, so what do we do once we have these fungi? So we describe biodiversity, first and foremost. We figure out who they are, we give them names, we add them to the global tally of known fungi. And we can do that very easily just here in Tucson without having to go very far. For example, I had an undergraduate student in the lab who found 3 new species of fungi in Gates Pass in 1 plant species. So just right there in the Tucson Mountains. This is how glorious they can be in culture. Is everyone feeling inspired? Right? Does that make you want to study fungi? Just kidding. Actually, that's beautiful. We don't often get those fruiting bodies, so that's really great. But the other thing is that we can actually find new species here on the U of A campus. So some of the species that we're working on describing now have been isolated from trees by Old Main. Okay, incredible to have this biodiversity resource here. All right, the next thing that we do is that we try to figure out how these endophytes interact with their hosts. So here I'm showing you my favorite plant of all time. This is the source of chocolate. Theobroma cacao is the tree. And the cool thing about these endophytes is that they are horizontally transmitted. So not like the grass endophytes, which are maternal plant to offspring, but horizontally transmitted. That means they infect plants from the air, spores that are drifting around the land and infect the plants. And so what we can do is we can grow seedlings that are sterile, and then we can infect them with endophytes and see what happens. And in this case, we even took it a step further. In the leaf that we see labeled pathogen plus endophytes, we have 1 pathogen and 7 species of endophytes. And that's a pretty happy, healthy leaf. Let's compare that to where we have the pathogen only. So we see that these endophytes can protect the plant against disease. And for chocolate, that's really nice because chocolate is actually a globally threatened crop. It's mostly grown by small landholders who don't have a lot of resources. And coming up with biological control methods that are inexpensive and use local positive endophytes is a really big step forward. So actually that's one of our projects, is helping small landowners grow healthy chocolate. And basically what we do is we get old rice, river water, and endophytes that we isolate from healthy leaves, spray that on the developing fruit in a given area, and that protects the fruit against disease. Okay. All right, the next thing that we look into is basically, are there interesting products that we can get from these fungi? And here I'm just showing you an example of a petri dish where that thing that looks like an egg yolk is one of our endophytes. And it is holding off by the production of interesting secondary metabolites a pathogen that wants to sweep over. Okay. So we look at what those metabolites might be and how we can use them to protect food. Now, it's not just food that we can protect, but of course, some of these metabolites have pharmaceutical applications. So basically, medicinal drug discovery. And in this case, we work with natural products chemists and with legal teams to ensure that we're following best ethical practices for discovering new drugs. That includes, for example, assuring that revenue goes back to host countries or communities. And we've been able to identify from endophytes more than 60 new compounds with activity against breast cancer and prostate cancer, cell lines, HIV, causal agents of childless disease, and so on. And so in some cases, we're able to publish that work based on endophytes we've isolated here at the U of A, okay, on our campus. Okay, again, outside of Old Main. So in the last little time that I have, I've given you just a flavor for sort of the questions and directions that we go. I just want to say that a lot of the work in our lab is really inspired by and driven by undergraduates. So Michael is one of our team members who is currently looking for new antibiotics from endophytes in medicinal plants. Okay? That's an action shot in the lab there for Michael. Victoria, who's so kindly introduced me, is taking that a step further and not just looking for how microbes can interact with each other, but can we discover through microbiomes of non-native plants like buffalo grass new tools for keeping those invasive species under control. Okay. And then finally, a student like Caroline is carrying that on a step further. She studies creosote and the endophytes in the roots. And it turns out that we think that those endophytes in the roots help creosote defend itself against competitors in the environment. All right, so last but not least, what we love to do is translate all of this into outreach. A lot of the work we do is really tractable. You can go out, get a plant, surface sterilize it, cut it up, grow out some endophytes, and do stuff with it. And so we've published a manual for how to do that at the high school and middle school level. It's basically like 72 pages of all the references you might need. And last but not least, I mentioned that we're taking a global perspective. So the first piece that I showed you was just our sampling in North America. But our goal is to really understand how different the endophytes are in each plant in each place. And basically it turns out that everywhere we go we find new species. So it's fabulous for us. And many of the habitats that we visited are shown here with photos. And I just grabbed this from my recent grant proposal to show you some of the places we've been. So we're just finishing a project called Go Life, in which we worked in Panama, Chile, South Africa, and Malaysia. You can also see stars across the top where we've worked in central Russia, eastern Russia, Alaska, and so on. Different endophytes in every place. And so the reason I'm highlighting that is that often when we think of a career in microbiology, we think of time in a lab with gloves and a lab coat and some dark room. And there is that. But also the great thing about microbiology and the environment is that we get to go out to amazing places. The far boreal forest in the north, tropical forests. This is a Yodé undergrad with me in Panama. It's one tree behind us. The beautiful volcanic grasslands of Chile. Post-fire sites here in the Chiricahuas. Sometimes studying organisms that are more spectacular than we could have hoped. This is a lichen, the largest known of its species. Deserts in other parts of the world, you name it. And so for me, that is really satisfying, and taking undergraduates with us to do that work is really meaningful to us. Okay. My closing thought is that so much of what we do is easily translatable. So we are going through basic microbiology and molecular ecology work in our lab. And we can translate that not just to study leaf endophytes, but for example, to study the microbes that live in nectar and how those influence pollination by insects. Okay? We also were really happy to work with a UBRP student for several years who helped us understand the oral microbiome of Cooper's hawks here in Tucson. He captured 30 Cooper's hawks. Let's just say that again. He captured 30 Cooper's hawks, swabbed their mouths, and helped us see that the pH of the oral cavity changes as the hawks age, and that relates to their susceptibility to a pathogen here in town. And then finally, we can translate this to a gratuitous sloth photo. And so one of the things about our work is that as scientists, we often are kind of in the trenches. We're doing really repetitive stuff that doesn't necessarily get a lot of glory while we're alive. But if it's good work, it's meaningful, and it means a lot, and it's really fun and satisfying to us. But occasionally, we work on something that gets picked up in the news. And so I'll just close with this translatable thought. So honestly, over beers one night, a few fellow scientists were talking about possibilities for future research in Panama. And someone said, you know, sloths are so interesting because they're covered with bacteria, cyanobacteria, with algae. I wonder what fungi are on there. And we all thought, I don't know, nobody's ever looked. It turns out that you can collect from the green slime on sloths the bunch of fungi that are related to endophytes, and they have a very high frequency of strains that produce anti-cancer metabolites. And so this led to all sorts of news coverage. And so for a while there, our whole team was like, oh yeah, you're the sloth people. We're like, no, we're the endophyte people, but we did a sloth thing. So sometimes our intended effect isn't necessarily what it was out to be. So with that, I hope I've given you just a flavor of the work in our lab and why we find fungi so interesting. Secondary metabolites, pharmaceutical drug discovery, enhancing agriculture, discovering biodiversity, and beyond. And with that, I'd be happy to take any questions. And thank you so much for your attention so late in the day. I really appreciate it.
So when it comes to horizontally translated fungi, how can you ensure that if you have one plant that has multiple pathogens or endophytes on it, that that one leaf is going to stay contained and you aren't going to have contamination from other horizontally translated?
Yeah, that's a great question. So the question is basically, given that it's like mayhem out there, it's basically horizontal transmission is happening all the time, right? And it happens with your microbiomes too. Over time, your microbiome becomes much more similar to that of your roommate, for example, right? You can still tell them apart, but you start to resemble each other on a microbial level, okay? Families do, dog owners do, all that stuff happens. So there's a lot of microbial mayhem. In our case, what we do is we will sample multiple leaves and get a sort of profile for an individual plant. And in general, these fungi are not sporulating out of the living tissue. So what's in there at a time is a snapshot of that diversity. And then from that, we can work back to figure out what is represented in that plant at that time. And then go back another time, another season, to ensure that we're getting a full profile. Does that answer your question? Great, thank you. Yes?
Can there be any like identifiers that transmit through like one organism eats similar and identify like it's transmitted?
That's a great question. Yes, there's been exactly one study of this, and they looked at a very glorious substrate, which was the fecal pellets of grasshoppers. And they showed clearly that when certain leaf tissue was eaten by grasshoppers, out of the fecal pellets would grow the endophytes that would sporulate and infect the leaves in it. So that's an example, the one example that we know of where that can happen, but I suspect it happens really frequently. Yeah, that's a great question. Thank you. Awesome. Thank you all so much.